Hello everyone, welcome back. So this is week 9 of the NPTEL course Experimental Biochemistry, and this course is coordinated by Professor Dasgupta from the Department of Chemistry and Professor A from the Department of Bioscience and Biotechnology, and I am Bias Mukherjee, a research scholar from the Department of Bioscience and Biotechnology, and in today's class we are going to talk about isolation of nucleic acids. Okay, so RNA and DNA isolation, the different strategies that are used, and these two topics are once again very important for your exams. Yes, a very good evening to you. Let's start with today's class. In today's class, we are going to talk about extraction of nucleic acids, extraction and purification of DNA and RNA. Okay, right. So let's begin. Okay, so we'll start talking about whether I'm feeling well or not. Yeah, I am better than last week, but still you'll see me coughing and all in between, but definitely better than last week. Okay, so let's start. Right, so now nucleic acids are mainly of two different types. One is DNA and the other one is RNA. Now there are a lot of different types of DNA and types of RNA, but for now we will look at only what is the basic difference between DNA and RNA, right? So the only difference is that DNA is made up of, there are many differences by the way. So DNA is made up of a special kind of ribose sugar, which is deoxyribose sugar, right? So what does that mean? So basically there is this H group instead of OH group that you can see in this figure, right? So instead of OH group in this position, there is this H group in DNA, and that makes it deoxyribose. And often DNA is seen in as two strands coiled together in the form of a double helix, and RNA is often seen as a single strand. But again, RNA assumes different kinds of structures, and it can form double-stranded configurations in different kinds of situations, right? So the different bases that are there in DNA and RNA, there is also a difference. So in DNA we have adenine, guanine, thymine and cytosine, right? But in case of RNA, instead of thymine there is uracil. So there is just one small difference between uracil and thymine, and the difference is that there is a methyl group in the 5th position in thymine, but then there is no methyl group in uracil. Now this small properties is what makes DNA the molecule that mainly is involved in carrying forward the information. Okay, so information is stored in one of the nucleic acids and it is carried forward in another nucleic acid. Now RNA is not very stable, and that is why genes store information, right? And genes are normally DNA. But RNA is not very stable, it gets degraded very easily. So we will talk about all the different enzymes that are involved in degradation of RNA, and why RNA is not the nucleic acid but the storage of genetic information. So we'll talk about that as well. Now initially there is this RNA world hypothesis where people argued that initially it was RNA that was formed, and initially all genetic material was possibly present only in that RNA. But later on, as evolution went by, DNA evolved to be the more stable molecule, and so now in our bodies we have all the genetic information stored in DNA. Okay, right. So now there are three parts of a nucleotide. So what is a nucleotide? Nucleotide is made up of the nitrogenous base. There is the sugar moiety, which is the ribose sugar, 5-carbon ribose sugar, and there is the phosphate group. Now these three units together make up the nucleotide, and nucleotides are linked together in a chain and they form RNA or DNA. Okay, so DNA is the storehouse of genetic material, while RNA participates in the transfer of this genetic material or genetic information to proteins performing various life processes. Okay, right. Now what is a nucleoside? This is again a very important, very tiny difference, but you need to be very clear with your basics. Okay, so nucleoside means that nitrogenous base is there and the sugar is there, but phosphate group is not there, it is missing. Only that part is called a nucleoside, but together all of the three are called a nucleotide. Okay, now we will talk about why RNA is so easy to get degraded. Right, so if you look at this structure structure of RNA, you will see that at this particular position, so RNA undergoes heat denaturation, and then denaturation under alkaline pH. Okay, and then there are these RNases. So let's see what happens. So RNA can be easily hydrolyzed in the following cases. One is heat degradation. So RNA can get degraded when heated at 65 to 70 degrees centigrade for 15 minutes, faster at higher temperature. Now, the interesting fun part is, if you remember PCR, polymerase chain reaction, you remember that DNA has to be heated at a temperature of around 90 degrees centigrade for the two strands to separate out. And then also it is intact, right? And that is a reversible reaction. When you decrease the temperature, it again forms double-stranded DNA. So there you understand how strong DNA is. But if you look at RNA, it completely gets degraded. Now, how is that degradation happening? See, there is this intermediate, like this, it is getting shortened basically. How is that? We will look at it. So, see, under alkaline conditions, this triggers RNA hydrolysis. It is stable at a pH of around 4 to 5. So, RNA is stable at a relatively acidic pH. Okay, so there is this like this OH minus, because in higher pH there will be this presence of OH minus, which high pH alkaline condition. Okay, so basically what is happening, there is this attack that is forming because of the presence of this OH group that is absent in DNA, and then 2' 3' cyclic monophosphate derivative is getting formed. Okay, now because of this, what happens, this monophosphate derivative is getting formed and the rest of the RNA is getting shortened. So that is how it is getting cleaved. Now another way is using RNases, which are enzymes, and these enzymes catalyze RNA hydrolysis. Okay. Now we will look at the RNases in a little bit more details. Now RNases, this topic you will find very interesting, because RNases are proteins that are very, very stable. Now we have talked about RNases in one of our previous classes, so you know about the catalytic activity of RNases and the active site of the RNases. So the interesting part about RNases is that they are very strong proteins. So even when you autoclave them once, that means you heat them at a temperature of 112 degrees centigrade for around 1 hour, they can still undergo renaturation, and they can once again form this functional protein that will come and cleave RNA. Okay, so RNases are released from their compartmentalized condition in cells upon lysis. They are ubiquitously present on our skin. Constant vigilance is required to prevent contamination of glassware, bench tops, reactants, reagents, etc. with RNases. Okay, so RNases are ubiquitous. They are present everywhere. There is no simple method to inactivate RNases because it contains a number of intra-chain disulfide bonds, which makes them extremely stable and resistant to inactivation by prolonged boiling or mild denaturant. They quickly refold after denaturation. So the structure of RNase A is one of the hardiest enzymes in common laboratory usage, and it cleaves single-stranded RNA. Right, so unlike DNAs, RNases do not require any divalent cation for their activity. So they are always active, right? And thus cannot be inactivated by using EDTA or any other ion chelators. So therefore, the best way to prevent problems with RNases is to avoid contamination in the first place. Okay. Now, RNases are so robust, but then the different, so how do you protect the RNA from these RNases? So for that, there are different kinds of inhibitors, right? So one of them is DEPC. So what is DEPC? This is diethyl pyrocarbonate. This covalently modifies the histidine residues which are present in the catalytic site of the RNases and inactivate them. Okay, but again, this is very toxic for the let's say you are using this for in vitro transcription and all, so you cannot use the RNase DEPC containing solution, because this is again toxic. So that is why you need to autoclave it twice so that this DEPC can be degraded. Okay. Another thing is autoclaving. So you have to autoclave twice to deactivate RNases. But sometimes even that does not work because it can sometimes get refolded. Another one is dry heating at 180 to 250 degrees centigrade. Also, you can use RNase inhibitors. Now, these are very expensive, but they are effective. And then you have to use gloves that are very, very sterile, okay, to prevent any kind of contamination. Okay. Now, we have a small MCQ. Try answering this. So, during plasmid DNA purification, RNA contamination is commonly removed by treating the sample with RNase A. Right? Which of the following statements correctly explains why this enzyme is suitable for this purpose? B. So it specifically cleaves double-stranded DNA by leaving the RNA intact. It cleaves RNA at phosphodiester backbone without requiring cofactors and remains active even after heat treatment. Okay, good. Let's see. Yes. So B is the right answer. Okay, great. Now we'll move on. Now, why do we need pure DNA and pure RNA? Okay, so DNA is required for a lot of different purposes such as genetic engineering, forensic studies, molecular diagnostics, therapeutics, whole genome sequencing, cloning, etc. For RNA, you need it for cDNA library preparation, gene editing, vaccine preparation, in vitro transcription, protein replacement therapy, molecular diagnostics, etc. So for all of these different purposes, you even know that for vaccine preparation and all COVID-19 vaccine, that was mRNA vaccine, right? So RNA is required for a lot of different purposes. So how do we purify this RNA and DNA? Right, so there are different methods. Today we will talk about all of these methods in details. So starting with Phenol-Chloroform. This is the most commonly used method and we will talk about this method in details. Okay, so organic extraction method that utilizes phenol and chloroform to separate nucleic acids from proteins and other contaminants. It involves multiple phases of extraction and requires careful handling of hazardous chemical. Then we have silica based columns. Now the interesting part about this is that silica is SiO2, right? So you know that this oxygen is there and to this silica, this is negatively charged. Now to silica, sodium binds. Now sodium will be there in the buffer. So as we talk about the different kinds of purification methods today, you will learn about this. So sodium helps to form this kind of, it allows silica to bind to DNA because normally DNA is negatively charged, silica is negatively charged. So sodium helps in neutralizing the charge a little bit so that silica can bind to DNA because DNA has a property to bind to silica. Okay, so it binds and helps in the purification of DNA, okay, and RNA. Now see, so silica, different silica membranes or beads are used for selectively binding to nucleic acid while contaminants are washed away. After binding, the nucleic acids are eluted from the column using a low salt buffer or water. Now popular kits include spin columns and 96 well plates. So very tiny spin columns are there in the kits and they are all silica columns. Okay. Then you have guanidine isothiocyanate. So this is something which is required for RNA isolation and this is very, very useful for RNA isolation. Right. So it utilizes guanidine isothiocyanate to denature proteins and solubilize nucleic acids followed by ethanol or isopropanol precipitation. So commonly used in protocols such as TRIzol. So TRIzol method we will talk about today, okay, in today's class. Then you have magnetic bead based DNA isolation. Then there are different methods such as Chilix, resin, etc. So you can read about this if you are interested. Plus there are also different methods such as anion exchange chromatography can be performed, organic solvent precipitation, okay, and then differential centrifugation, this we will talk about again, and alkaline lysis is something that we will talk about in details, and enzymatic digestion can also be done. So enzymes such as proteinase K, lysozyme, this can be useful for degrading the proteins, and enzymes such as RNases are useful to degrade RNA. So you get only DNA. Okay, right. Now we will start with one of the very important methods, and that is cesium chloride density gradient centrifugation. Okay, so this method is based on the phenomena of buoyant and specific density, buoyancy and specific density. Right, so cesium chloride is an extremely dense salt. So if you remember the position of cesium in the periodic table, you will know that it is down below, and that is why it is very, very heavy, right, compared to sodium, calcium, etc. So cesium chloride is extremely dense, and when the solutions of salt are subjected to centrifugation at a very high speed, typically 100,000 rpm, cesium chloride sets up the concentration gradient, where it is highly concentrated at the end or side of the centrifuge tube. One second. Okay. Yeah. So, right. So basically, what happens is where it is highly concentrated at one end or side of the centrifuge tube that it is in, and much less concentrated at the other. So if you can see this pattern, from there only we will understand. So if the centrifugation process is ended gently, the centrifuge slowing down slowly, the denser solution, that is with higher cesium chloride concentration, will settle at the bottom of the tube. So all the solutions that are having higher density will settle at the bottom, and then gradually it will be a gradient, and the one with the least density will settle at the top. Okay. Now, other molecules dissolved in the solution will separate themselves according to how dense they are, with the denser molecule going towards the bottom of the tube and the less dense molecules going towards the top of the tube. When DNA is in presence of a molecule called ethidium bromide, it will bind to it and become fluorescent under UV light, right? This you know. Ethidium bromide also adjusts the density of DNA so that it moves towards the center of the tube upon centrifugation. Contaminants will move to different positions and therefore will be separated from the DNA, which can easily be seen and recovered since it will be fluorescent. Okay, subsequent steps will be used to separate the DNA from the cesium chloride and ethidium bromide. This technique is particularly effective for separating plasmids, which are small DNA strands that bacteria use to share information on antibiotic resistance with one another, and mitochondrial DNA and other such specific DNA molecules that bind to characteristic amount of ethidium bromide and therefore will have unique densities. It is used to separate mitochondrial and plasmid DNA also. So these nucleic acids are arranged in highly coiled, often called supercoiled. You know what supercoiled is, right? So supercoiled is very covalently closed and circular. That makes it supercoiled. So circular patterns DNA that is not supercoiled, for example genomic DNA or chromosomal DNA, binds to different amount of ethidium bromide and therefore have different density, making it easier to separate them from the plasmid DNA. So if you look at the figure, you will see that there is protein, then comes your chromosomal DNA, and then comes supercoiled DNA because it is more dense compared to linear DNA, and finally there is RNA. So that is how you can actually extract the different types of DNA that you want to isolate. Okay, now there are two very interesting things. One is GITC mediated nucleic acid extraction, and the other one is CTAB mediated extraction. So let's look at this. So guanidine isothiocyanate. So what is this? An aqueous solution of salt guanidine thiocyanate when mixed with solvent phenol and chloroform allows for effective purification of RNA through minimal number of steps. When the samples are ground up and mixed with chloroform, phenol and sodium acetate and guanidine thiocyanate and then subjected to centrifugation, the salt solution will separate from the solvents, providing an upper aqueous solution and lower organic phase. Okay, so RNA will remain in the aqueous solution while the proteins and nucleic acids and other contaminants will go into the organic phase or in the interface between the two phases. So if you look at the figure, you will see that guanidium is applied and phenol is also provided. Then what is happening? RNA, DNA, protein, all of these are present, and then you're adding chloroform. then what happens is the phase separation happens. So the upper layer contains RNA. In between, at the junction of the two layers, there is protein, and in the organic phase, there is your DNA. Okay. Now what happens? You precipitate it. So you add isopropanol. Then what happens? RNA forms a pellet, and the rest of it is just solution. Then you desuspend it and you get whatever you want. Okay, so now another method is CTAB. So what is the CTAB thing? So cetyl trimethyl ammonium bromide nucleic acid extraction. So CTAB is basically mostly used for plant DNA isolation. Right, so this technique is commonly used for plant samples as well as their parts such as grains, seeds and leaves. So it is used for many food samples. Nucleic acid and some other polysaccharides are insoluble in solution of 2% CTAB at high pH. So neutral polysaccharides and proteins are soluble. This provides an easy way to remove many difficult plant contaminants prior to purification. Right, so like this is a positively charged entity, and a lot of like you see that neutral polysaccharides and proteins are soluble, but nucleic acids are insoluble. So you're basically precipitating the nucleic acid, and then you can isolate the nucleic acid from this solution. Okay, right. Now we will look at this. So the plant sample is there. You grind it in liquid nitrogen. Then you add CTAB, and then at 65 degrees centigrade it is kept and then spun at 15,000 G. And then chloroform is added. So chloroform plus isoamyl alcohol in the ratio 24:1. Remember the ratios also. Okay, so 24:1, that is the ratio, and then it is chelated on ice. Then again it is centrifuged and 100% ethanol is added for the precipitation purpose. Right, so for precipitation this is added, and then you see that DNA is getting precipitated and all the other things, for example all the polysaccharides and proteins, they are all in the soluble layer. Then you add ethanol, you throw all the supernatant and you add ethanol again, 70% ethanol this time, and then you elute it using the elution buffer. Okay, and you get pure DNA that way. So this is the procedure. Now we are going to talk about something that a lot of you might have done in the lab already. Okay, and that is isolation and purification of nucleic acids, and this step is mainly used for bacteria. Okay, so transformed E. coli DH5 alpha cells, hopefully harboring your plasmid of interest, were plated on LB agar plates, supplemented with 50 microgram per ml kanamycin to maintain the plasmids in the cell. This is the antibiotic to ensure that the plasmid is held by the cell. Why? Because the plasmid contains the resistance gene for this antibiotic. Otherwise, the bacteria will die. Right? So now plates were incubated overnight at 37 degrees centigrade and one individual colony. So it is thought, it is believed that every single colony is getting formed from one single cell. Okay, so that tiny cell is called colony forming unit. So that means all the entire colony population has the same genetic makeup. That is what is considered. So if we pick that colony, so see what is happening. Now one individual colony, that is white circle highlighted by box, was picked and placed in a tube containing 5 ml liquid LB media supplemented with 50 microgram per ml kanamycin, and it is grown overnight. It becomes turbid. Okay. Now what happened? The tube containing your cell suspension was incubated overnight at 37 degrees centigrade, and this steps allows for propagation of a large number of cells containing your plasmid of interest. So these cells are theoretically genetically identical as they were cultured from a single colony. Okay. Now prior to the alkaline lysis protocol, E. coli DH5 alpha cells containing your plasmid must be harvested from the liquid LB to remove the excess media from the system. This is done by centrifugation. So it is spinned basically, and all the cells are pelleted down at the bottom. All the media is on the top. You throw the media. So this is done by centrifugation for the separation of the liquid media that is supernatant from the cells that is pellet. Bacterial cells are very tough and they do not typically break open by centrifugation. Okay. So now what you get is this cell extract. Now something is done. So see what's done. So this is again organic purification. Okay, phenol-chloroform based purification. So for the organic method, that is phenol-chloroform based method, this uses SDS, proteinase K and RNase A for the enzymatic digestion of proteins, RNA and non-nucleic acid components. So a mixture of the ratios you have to remember. Okay, ratios are very important. Phenol, chloroform and isoamyl alcohols. Phenol, chloroform, isoamyl alcohol. 25:24:1 in the ratio. Then it is added to promote the partitioning of the lipids and cellular debris into the organic phase, leaving isolated DNA in the aqueous system. Okay, so when it is centrifuged, you see that the aqueous part containing the nucleic acid is there. In the interface, there is coagulated proteins and the organic layer contains phenol. Okay, so next see what happens. In this extraction method, isoamyl alcohol is the anti-foaming agent. It helps in reducing the foaming between the interface and prevents emulsification of a solution so that the layers can be distinctly formed. Okay. Right. Now another important thing is you will have to know the exact purpose of phenol and chloroform. Okay, so what is the exact reason chloroform and phenol are used? So that you will find out. Okay, so chloroform, as far as I remember, it is mainly used to make phenol very dense and make sure that it settles at the bottom, because phenol is normally very light. Right, so how will you make sure that the organic phase goes at the bottom? So for that, chloroform is added. Right. Now alkaline lysis. So you remember that pellet that was formed in the previous slide. Now you take that pellet. So what do you do with the pellet now? You can go for alkaline lysis. Okay. So plasmid is an extracellular DNA molecule different from the chromosomal DNA, which is capable of independent replication. The term plasmid was first used by the American molecular biologist Lederberg, and plasmids vary in size from 1 to 1000 kilobase pairs. Plasmids are mostly circular and they are double-stranded. Plasmids are found in a wide variety of bacterial species, but mostly they are found in eukaryotic organisms. For example, yeast is given as an example. Now, plasmids are found in bacteria also. Okay, and plasmids provide different functions. For example, antibiotic resistance. Plasmid can help in propagation of different genetic information. They are useful for like a particular protection from, okay, they can be used for production of some kind of toxin. Okay, so there can be different kinds of plasmids which are mediating different kinds of resistant properties and then production of a specific substance. Right, so all of these different things that are assisted by plasmids, and that is why the bacteria carries them. Okay, now see, so pelleted bacteria comes, you have the pellet, you have the supernatant. Now what do? Add lysis buffer followed by neutralizing buffer. So what is lysis buffer and what is neutralizing buffer? So we have everything in the next slide. All the different compositions are there in the next slide. Okay, centrifuge to pellet the protein precipitate. So protein is getting precipitated. Transfer the supernatant to the spin column. You can see that this blue thing is the spin column, and then the DNA gets stuck to the spin column, the rest of the fluid flows by and then it is eluted. Okay, and you get the pure plasmid DNA. Now exactly what is happening, we will see that right now. Okay, this is a very important concept, you have to understand this. So you can see that the chromosomal DNA is massive, it's huge, and it is normally, it is not supercoiled, right? So it is really huge, and what happens is once it gets denatured, it is very difficult to renature it. Okay, so see, alkaline lysis in combination with the detergent SDS has been used for more than 20 years to isolate plasmid DNA from E. coli. So exposure of bacterial suspension to strongly anionic detergent at high pH opens up the cell wall, denatures the chromosomal DNA and also the plasmid DNA and proteins, and releases the plasmid DNA into supernatant. Although the alkaline solution completely disrupts base pairing, now the interesting thing is both chromosomal DNA and the plasmid DNA, they get denatured, right? But the plasmid DNA, even though it is denatured, the strands are still close by because it is having this supercoiled configuration. So the strands are very, very close by. So what happens when you remove that high like pH and everything conditions, then once again they come close by and they will form the hydrogen bonds, and it will once again become renatured DNA, right? But the chromosomal DNA, it is a complete mess now because it has opened up and it does not know how to form the H bonds and all that. So it is kind of taking a lot of time to get renatured fully, but your plasmid DNA immediately renatures, and that is how it becomes soluble and we can separate it out, whereas this one remains insoluble because it is getting pelleted, right? So now we will look at it in a little more detail. So that is the entire concept of separating plasmid DNA from genomic DNA. Okay. So although the alkaline solution completely disrupts base pairing, the strands of the closed circular DNA are unable to separate from each other because they are topologically intertwined. This is your supercoiled plasmid DNA. As long as the intensity and duration of exposure to OH minus is not too great, the two strands of the plasmid DNA fall once again into so they once again fall into one another, and when the pH is returned to neutral, they again form this double-stranded configuration. During lysis, bacterial proteins, broken cell walls and denatured chromosomal DNA become enmeshed in a large complex that is coated with rhodesyl sulfate, and these complexes are efficiently precipitated from the solution when the sodium ions are replaced by potassium ions. Okay, and since it becomes insoluble, what happens is only your plasmid DNA is soluble now, so you pick that up and the rest of it is insoluble white mesh, and you throw it off. So after denatured material has been removed by centrifugation, the native plasmid DNA can be recovered from the supernatant. Okay, now we will look at this process in a lot more details. Okay, so there are these 3 steps. We will look at them one by one. Okay, so first thing, you got that pellet. You remember the bacterial pellet came. You threw away the supernatant containing the LV media. Fantastic. Now what do? Now we dissolve this in a resuspension buffer. So what is this resuspension buffer? It is made up of TRIz and EDTA. TRIz has buffering capacity, pH of around 8. Okay, and EDTA, it chelates the divalent cations. Now why is that necessary? Because remember, DNases need divalent cations to act. So DNase, if it becomes active, it will chop off the DNA that you have. So we need EDTA to remove all of those divalent cations. So these cations typically function to stabilize the cell wall and maintain DNase activity. Okay, so glucose. We also need glucose in the solution. Why? To maintain osmotic pressure, and RNase A is there to degrade the RNA once the cells are lysed. Okay, next thing is lysis buffer or solution 2. So what is lysis buffer? We need to break open the cell. How do we do that? We use NaOH. So NaOH is used to break open the cell wall and disrupt the hydrogen bonds between the DNA. Okay, so now both the DNA are getting denatured. So the result is result in DNA denaturation into single-stranded DNA, and SDS is also there, and SDS detergent stabilizes and solubilizes the cell membrane and denatures the proteins. Okay, that is the role of SDS. Now we have neutralization or precipitation step. So what is happening here? Buffer contains potassium acetate of pH 4.8 to 5. Serves to decrease the alkalinity of the preparation and reform the hydrogen bonds between the complementary single-stranded DNA bases. In this step, we must consider the small size of the plasmid DNA in comparison to the chromosomal DNA. The plasmid double-stranded DNA is readily dissolved in the solution, but large genomic DNA is tangled in a meshwork of denatured cellular proteins and other cellular debris, mainly driven by hydrophobic interactions. So this results in the white fluffy precipitate. Okay, so this is the entire principle of plasmid DNA purification. Okay, great. So now we will talk about isolation of RNA. Okay, this is again very interesting. So there is something called TRIzol. What is TRIzol? There are 3 ingredients. So these 3 ingredients are helping in purification of RNA. Okay, so what are they? So TRIzol is a monophasic solution of phenol, GITC and chloroform. 3 ingredients. Okay, in a pH of 7. Water saturated phenol and aqueous solutions of GITC, sodium citrate, lauryl sarcosine and DME. It is used for the isolation of RNA. It results in the formation of 3 phases. The aqueous phase contains RNA, interphase contains DNA and organic phase contains proteins and lipids. Okay. Now the low acidic pH of TRIzol controls separation of RNA and DNA from protein, while a high pH can cause RNA and DNA to be isolated together. Okay, that is why in the plasmid isolation protocol you saw RNase A was there to prevent any RNA in the product. Okay, now guanidine salt serves as cheotropic agent and denatures the proteins, and the phenol that is indicated in pink color is an organic compound which is used to extract the nucleic acids and proteins. Okay. Now after solubilization and homogenization of the samples in TRIzol, the RNA, DNA and protein are differentially extracted by addition of a phase separation reagent, that is Phenol-Chloroform. The solution separates the RNA away from the DNA and protein into different layers that we can see in the figure. Subsequently, the RNA in the upper aqueous solution is collected by alcohol-based precipitation. So you saw in that figure that precipitation with ethanol was performed. It is air-dried and resuspended in the final sample buffer. Okay, so that is what is done. Now we will try to solve a few questions. Okay, so you can answer in the chat box. Let's start with the questions. So during RNA extraction, so during RNA extraction, GITC is commonly used. What is its primary role? Selectively precipitates RNA, denatures proteins and inactivates RNases by disrupting hydrogen bonds, chelates divalent cations required for DNase activity. B. Great. What about the next question? In phenol-chloroform extraction, after centrifugation, DNA is typically found in which phase? Okay. So this you have to remember, in which kind of technique where DNA is found. Okay. So this is very important. It's very important for you to remember because these kind of questions will be there in the exams. So yes, that is correct. So B and C. Okay, it's found in, DNA is found in the aqueous phase in the phenol-chloroform extraction method. Okay, great. Right. Now, the next one is the use of CTAB. So how will you remember whether DNA is found in like organic phase or aqueous phase? Remember, if it is in organic phase, then you cannot recover it. So if that method is used for separation of RNA, then RNA will never go into the organic phase. But if that method is used for separation of DNA, then DNA will always be in the aqueous phase. That is how you will remember. Okay, right. The use of CTAB in plant DNA extraction is especially useful because it hydrolyzes RNA, removes polysaccharides by forming insoluble complexes, decreases solubility of DNA in ethanol. It acts as DNase inhibitor. During plasmid DNA isolation by alkaline lysis, chromosomal DNA is separated from plasmid DNA because plasmid DNA is linear and small. Chromosomal DNA denatures faster than plasmid DNA. Plasmid DNA renatures correctly while chromosomal DNA aggregates irreversibly. Plasmid DNA is resistant to alkaline pH. Okay, which one do you think is the right answer? B and C. Good. Yeah, correct. So we'll look at the right answers. Yeah, that is true. B and C. But then remember that chromosomal DNA does not aggregate irreversibly. It aggregates reversibly. So if you leave this solution for 2 days or so, 2 to 3 days, maybe sometime it will once again renature back. Okay, but then that is not practical, and that is the thing that we exploit for isolation of this supercoiled plasmid DNA. Okay, right. Now we have 2 more questions. Try answering them. In cesium chloride density gradient centrifugation, plasmid DNA is separated based on molecular weight, charge, Boyne density differences due to supercoiling, and sequence specificity. TRIzol contains phenol and guanidine salts. The key advantage is selective isolation of DNA only, simultaneous extraction of RNA, DNA and proteins from a single sample, exclusive removal of lipids, prevention of DNA denaturation. So which one do you think is the right answer for them? Okay, so C and B, yeah, good. C and B are right answers, right? Okay. Right. Now we have 3 more questions here. So in phenol-chloroform extraction, if phenol is adjusted to acidic pH, which molecule preferentially partitions into aqueous phase? Acidic pH. Okay. Now, why is CTAB sometimes preferred over SDS in plant DNA isolation? Okay. In TRIzol extraction, the role of GITC is most closely associated with DNA precipitation, RNase inactivation and protein denaturation, phase separation, GITC, okay? And lipid solubilization. What about CTAB? CTAB is non-ionic. CTAB binds to polysaccharides while SDS does not. SDS cannot lyse cells. CTAB degrades proteins more efficiently. All are B. Okay. Let's see. Let's look at the right answer. Yes, very good. All are B. Okay. So when the pH is 4.5, so remember RNA is more stable in lower pH. For DNA, the pH is relatively higher, right? So in phenol extraction, if the phenol is adjusted for acidic pH, the molecule that is preferentially partitions into the aqueous phase is RNA. Now, why is CTAB sometimes preferred over SDS in plant DNA isolation? CTAB, remember, it is positively charged, right? So, if it is positively charged, the polysaccharides are negatively charged. So, definitely they are going to come and bind, right? So, CTAB binds to the polysaccharide and SDS does not, because SDS is also negatively charged. Correct? Okay. In TRIzol extraction, the rule of GITC is RNase inactivation and protein denaturation. Because phase separation, remember, phenol and chloroform are doing, right? So phase separation is not the work of GITC. Okay. Now we have something very, very important, and that is absorption. Okay. Now you will get questions from UV visible spectroscopy. So this you have to remember. Okay. So what absorbs where? So at 230 nanometer, there is absorption by salts. At 260 nanometer, there is absorption by nucleic acids. At 280 nanometer, there is absorption by aromatic protein rings. Okay. Protein contaminated with nucleic acids will also have absorption, but different ratios would be there. Now, another very important thing is, besides the aromatic groups in proteins, what about the like the protein backbone, and what about cysteine? So all of these different molecules, they can also contribute to absorbance. So you will have to learn about those as well. Okay, so you can actually read about that. So what about the backbones? Whether the backbone absorbs and at what nanometer? So that you will have to find out. Okay, now thumb rule. So 260 by 280 for pure DNA it is 1.8 and for pure RNA it is around 2. 260 by 230 is 1.8 for pure DNA and for pure RNA it is more than 1.8. So more than 1.8 for both. Okay. If this 260 by 280 ratio is less, that means the absorbance at 230 is more. That means there is a lot of salts in that sample, right? And if the 260 by 280 ratio is higher, that means there is contamination from proteins that we can see in this figure, okay, in the table. Okay. Now a ratio of 1.8 is generally accepted as pure for DNA. Ratio of 2 is generally accepted as pure for RNA, but why? That is because RNA, the bases are exposed. So they will absorb more. But in DNA, the bases are stacked. So they have lesser absorption. Correct? So if DNA has been chopped, then it will have more absorbance. The 5 nucleotides that comprise DNA and RNA exhibit widely varying 260 by 280 ratios. Now another important factor is that the absorbance of uracil and absorbance of thymine is very different. Okay, so absorbance of uracil is 4 but absorbance of thymine is 1.47. So for the same concentration of RNA and DNA, RNA will absorb a lot more compared to DNA. So that is something that you have to remember. Okay. Now, RNA will typically have higher 260 by 280 ratio due to higher ratio of uracil compared to that of thymine. So that is something very important. The actual ratio will depend upon the composition of the nucleic acid. So concentration of DNA sample in microgram per ml is equal to 50 into A260 into dilution factor. Okay. This is how you calculate concentration of DNA. There will be questions on this also. So it's good if you remember. Okay. Now we have UV spectrophotometric measurement of pure RNA. Okay. Now see, pure RNA will have 260 by 280 ratio of 1.8 to 2. A ratio of less than 1.8 indicates potential contamination, likely due to mixing phases when removing the upper aqueous phase of the TRIzol separation. This method does not discriminate between DNA and RNA. It is advisable to treat RNA samples with RNase-free DNAs to remove the contaminating DNA. Okay. A260 by 230 ratio should be above 2. Remember that. So a low 260 by 230 ratio shows contamination with the washed solutions, cheotropic salts, phenols or proteins, likely due to inefficient washing. Okay. Right. Now we have questions. Let's try to solve these questions. A nucleic acid sample shows the following ratios. A260 by 280 is 1.55 and 260 by 230 is 1.1. So the ratios are low. Okay. Which of these are the most likely interpretations? Pure DNA samples, protein contamination only, contamination with phenol or cheotropic salts along with protein, RNA contamination only. C. Good. Now, why do nucleic acids absorb strongly at 260 nanometer? Why? Peptide bond transitions, aromatic amino acid side chain, C is pi-pi transitions in nitrogenous bases. Correct. Okay. So yes, the right answer is C for both. Right. Now, during RNA extraction using TRIzol, improper phase separation leads to carryover of the interface material. So what spectral change is most expected? So if you remember, at the interface there was DNA, right? So now what will you see? Increase in 260 by 280, decrease in 260 by 280, increase in 260 by 230 and no change in the absorbance ratios. Okay. Extraction of the acidic phenol results in RNA in aqueous phase and DNA in organic phase. B. B is 260 by 280 ratio decreases. Okay, let's see. And then we have extraction with acidic phenol results in RNA in the aqueous phase and DNA in the organic phase. If this if this RNA shows A260 by 231, the contamination is most likely phenol and cheotropic salts. Okay. So we will look at the right answer. Yes, the right answer is B and C. Okay, so let's go back to this TRIzol once again. So yes, this is what was there. Aqueous phase, interphase and proteins and lipids. Okay. Right. Now we have some more questions. Question on RNA stability. Now RNA is chemically less stable than DNA. Why? Because of hydroxyl group at the 3' position, presence of hydroxyl group at the 2' position, presence of ester bond at the 3' position. RNA has uracil while DNA has thymine. Okay, presence of hydroxyl group at the 2' position. Yes, that is correct. So presence of hydroxyl group at the 2' position is what makes it so susceptible to degradation, right? And this cyclic intermediate is getting formed because of that. 2' OH is the culprit. So RNA is made up of ribose carbohydrates rather than deoxyribose. It is more unstable than DNA because of the presence of hydroxyl group, which renders it more susceptible to hydrolysis and disruption. In general, RNA is unstable due to chemistry. The absence of this hydroxy group in DNA is also known as 2' deoxyribonucleotide ribonucleic acid, and that is significant because RNA has a typical 2' hydroxyl group. The phosphodiester bond is unstable and prone to nucleophilic assault and self-hydrolysis. Okay. Now, isolation of DNA from plant sources possesses challenge due to the presence of high amount of RNA, high amount of protein, presence of polysaccharide and secondary metabolites, and low amount of DNA. What do you think is the right answer? Why is plant DNA isolation so difficult? Very simple question. C, exactly, yes. Because polysaccharides and secondary metabolites are there in plants, right? And these are not there in bacteria or anything else. So see, the cell wall compositions, plant cell wall is made up of, cellulose, hemicellulose, lignin. Breaking these barriers are very difficult. Next, you have the secondary metabolites. There are so many. There are phenolics, polyphenols, etc. These compounds can co-precipitate with DNA during extraction, leading to impurities in the DNA sample. Then nucleases. Now, plant tissues contain high level of nucleases which degrade the DNA rapidly. Proper inhibition is very important. Okay, and polyphenols and polysaccharides. So they form complexes and makes the DNA purification difficult. So you need to remove all of them, and that is why you have this full CTAB extraction process where all of this is removed. Okay. Now there's another question. The function of EDTA during cell lysis is act as metal chelating agent, denature protein, destabilize cell wall, all of the above. A. Okay. Yes, that is true. A. And that is the structure of EDTA. So EDTA is a chelating agent, divalent cations such as Mg2+ and Ca2+. They are used by DNases which break down DNA, and the DNase activity and integrity of the bacterial cell wall requires all of these different cations. So EDTA chelates divalent ions in solution and prevent DNase from damaging the plasmid. Destabilization of the cell wall is also done by it. Okay. So if that would be an MSQ, maybe you could do all of the above. No, I did not write it. I took it from some internet source. So I did not write it. Okay. Right, so we'll go on. But the right answer is chelating. Okay, so that is its primary function. Now there is this MSQ or nucleic acid isolation. What is the functions of NaOH in lysis buffer? So denatures genomic and plasmid DNA, NaOH in lysis buffer, right? So degrades the RNA, breaks down the cell wall, removes protein from the cell lysate. What is the function of NaOH? It does a lot of things. Try to recall. What is the role of SDS in lysis buffer? This is very straightforward. A, okay, and D. For the second question, it is D. That is correct. Okay. But for the first one, it is A, C, and D. So we will look at the right answer. So A and C. Yes. Acha, yeah, right. So A and C. Because NaOH also helps in breaking down of the cell wall, right? And removing protein from the cell lysate. This it is not doing. This is done by SDS. And NaOH's main role basically is denaturation of plasmid and genomic DNA. But it also helps in breaking down the cell wall because it is made up of proteins. Okay. Now, lysis buffer contains NaOH which breaks down the cell wall and disrupts hydrogen bonds between the bases. This results in DNA denaturation into single-stranded DNA. SDS destabilizes and solubilizes the cell membrane, denatures many other cellular proteins. Okay. So isolating plasmid DNA from genomic DNA is based upon conformation, charge difference, size difference, cellular location. Which one do you think is the right answer? A and C. Okay, let's see. Yeah, that is correct. So A and C are the right answers, right? So phosphate buffer contains potassium acetate of pH 4.825, which serves to decrease the alkalinity and of the preparation and reform the hydrogen bonds between the complementary single-stranded DNA bases. In this step, we must consider small size of the plasmid DNA in comparison to the chromosomal DNA. The plasmid double-stranded DNA is readily dissolved in the solution, but the large genomic DNA is tangled in a meshwork of denatured cellular proteins, while other cellular debris, mainly driven by hydrophobic interactions, they remain as a white fluffy precipitate in a denatured and precipitated condition, right? Whereas the plasmid DNA denatures because of its small size and conformation. Now, coming to conformation, I'm sure you remember this. Now, plasmid DNA may appear in one of the 5 different conformations. It can be nicked and open circular, it can be relaxed circular, it can be linear, it can be supercoiled, it can be supercoiled and denatured. So, these are the different conformations. Normally, intact plasmid DNA has supercoiled conformation, and that is why it is easy to remove it, to renature it back. Okay. Right, so we had talked about this in one of our previous classes, if you remember, where we were talking about the motility of different DNA conformations on gel, right? So I will not go through it anymore. So now we have another question, try to solve this. Elution buffer contains, what are the components of elution buffer, and why when neutralizing buffer is added after lysis to the cell lysate, then what happens? Okay, try answering this. A, D. So elution buffer, besides TRIz, it also contains EDTA. Okay, and neutralizing buffer. So let's see. Yes, so elution buffer contains both TRIz and EDTA. Why EDTA? You know this. You know the right answer. Answer in the chat box. And neutralizing buffer is added after lysis. Why? Chromosomal DNA gets tangled and precipitates out. Plasmid DNA anneals and remains in solution. Protein and SDS form an insoluble precipitate. So yeah, correct. Right. What about the first one? So EDTA is added. Why? Because EDTA chelates Mg2 plus and Ca2 plus, and Mg2 plus is required by DNases for cleaving DNA. So even if there is any DNase in the solution in the water that you're providing, to ensure that it is not functional, we add EDTA. Okay. Right. So now we have another question. During extraction of the plasmid DNA, after the addition of neutralization buffer, which biprecipitate is formed due to the formation of insoluble potassium dodecyl sulfate salt, soluble potassium dodecyl sulfate salt, soluble potassium acetate. So very straightforward question. If it is white and if it's a precipitate, then what is it? Exactly, it is definitely insoluble. Correct? Yes. So it is definitely insoluble, fluffy white precipitate containing large genomic DNA tangled in a meshwork of denatured cellular proteins and other cellular debris. And if you remember, buffer contains potassium acetate, which is soluble, and final one contains potassium dodecyl sulfate, insoluble salt. We have two more questions here. DNA bound to silica membrane can be eluted by addition of. To wash the DNA bound to silica membrane, which of the following reagents are used? TRIz, deionized water, ethanol and isopropanol, and detergent. Okay, so you're saying that it is C. Low ionic strength buffer. Actually, let's see. Let's look at the right answer. Okay, A and C, and this is C. Okay, let's see which is the right answer. So for the first question, yes, A and C are the right answers, right? So load supernatant from the lysis step onto the spin column. Centrifuge for 1 minute. Discard the liquid from the collection tube. Okay. Add wash buffer. So centrifuge for 1 minute and discard the liquid from the collection tube. And then place the purification column into fresh microcentrifuge tube where you will collect it. Add elution buffer, that is TE, to the center of the tube. Incubate for 1 minute at room temperature and centrifuge for 2 minutes to elute DNA. This is what is done. And it can be eluted using nucleus-free water. You can either use water or you can also use EDTA containing buffer, okay, which is the elution buffer. So elution buffer has TRIz and EDTA. TRIz pH is 8. Okay. Now, the next one, ethanol and isopropanol are used for washing. So when you wash, what happens? It gets precipitated. DNA gets precipitated and then only the pure DNA is there. And then when you elute it, it once again solubilizes and gets eluted. Correct? Right. Now the DNA is stuck on the silica more extensively. You want to remove the salt so that it doesn't mess up things later on. So the wash conditions involve ethanol. Now ethanol is important because it dissolves salts but not DNA. So you can wash the salt easily. But also interferes later. So make sure that you spin twice to remove excess. That is why there is something called a dry spin. So what is a dry spin? So this ethanol is there in the wash buffer. Okay. So after your neutralization buffer, there is this wash buffer. Wash buffer has a lot of ethanol. And ethanol, it is very important because it removes the salts, but it will not remove the DNA. And ethanol is very important to be removed because later on it will obstruct in the downstream processing. And also if you're running it on a gel, if it is in a buffer, ethanol will obviously float up higher and your DNA will be floating up higher, and it will come out from the well. It will not go into the well of the agarose gel. So that is why it is very important to remove ethanol. Okay, and that is why there is something called dry spin. So you don't add anything, you just spin the column once again in the centrifuge for 1 minute to make sure whatever is there in the column that comes down, and you throw it before the elution step. So low salt and higher pH, silica membrane becomes negatively charged. That is why TRIz of 6 pH was not used, but TRIz of 8, TRIz pH 8 was used, so that the silica membrane becomes negatively charged like the DNA, and they repel, and DNA gets coated in water and comes out. Okay. To wash the DNA bound to silica membrane, which of the following reagents are used? So ethanol or isopropanol is used for washing. Now we have 3 more questions. Try answering. Quality of the purified plasmid DNA can be checked by ACC, agarose gel electrophoresis, UV spectroscopy, and SDS page. Okay, these are MSQs, so more than one right answers can be there. Neutralization buffer contains which salt? And then you have in agarose gel electrophoresis, plasmid DNA runs ahead of the linear DNA. Why? So the first question is an MSQ, the other 2 are MCQs. Okay, that is a hint for you. Now try to answer. Very straightforward. One is B and C. Yeah, correct. And 2 and 3, these you know. 2 and 3 are very, very straightforward, right? Very simple. C. Yes. So second question is C. What about the third one? Third one is also C. Great. That is correct. Yeah. So we have B and C as the first one, and we have C in the second one and C in the last one. Okay. Great. Now we come to the last question of today. RNase A degrades RNA that is present in the cell lysate. What will happen if RNase A treatment is not done during DNA isolation? So DNA will get degraded, RNA will not be removed, as both DNA and RNA can absorb at 260 nanometer. Quantification of pure isolated DNA will be problematic. Correct. So yeah, B is the right answer. You will not get the right reading for pure DNA. Correct. So that's it. These are your references, and thank you for paying attention to today's class. Thank you for attending all my classes so consistently. Thank you so much. Okay? Yeah, sure. Please ask. Which exam I gave? So first you will have to qualify GATE. Once you qualify GATE, all the IITs, they will release their applications for PhD. You will have to fill in the applications, and then in our case, there was this, we had to come here and there was this exam. Okay, it was not online, it was offline. So that offline exam, so I filled the application, that was the first checkpoint, and then some of the applications got accepted. Then they called us and we came here, and then there was this written test. Now it has become computer based test. When I had joined, it was written, so I cleared the written test. Then in the third round there was this interview. Now also it is similar, so after this computer based test there is an interview. So in the interview all the panelists will be there. So whatever is your stream, in one room there will be professors from that particular stream and related few streams. They will ask you questions, you will clear you'll answer their questions. So how many years of PhD are you doing? 3.5 or 5? So PhD at IIT is 5. PhD everywhere is normally 5 years. Okay, you get fellowship for 5 years only. And sometimes it takes more time, sometimes it takes less time. So normally for engineering streams it takes less time. But for biology it will not take less time, unfortunately. It will normally take around 5 years. Some places it takes more time. Depends upon your project. Okay, depends upon a lot of different factors. So yes, that is it. So once you qualify the interview, if they select you, they select you, right? So it depends upon the lab and depends upon whether there are opening positions in the lab or not. So one thing that I would suggest to you people is you can actually mail the professors beforehand that I am interested in your lab. That often works very well. So in my case, I did not know the professors. I hadn't mailed. But it's often advisable that you mail the professors beforehand. They would be very interested to know that you are interested in their work. So then they'll be very interested to actually speak to you and to take you up. Okay, so it's good if you have your own fellowship, but I did not have my own fellowship, so that is also okay. So if you clear any of the exams, for example CSIR-NET or DBT or ICMR, you will have your own fellowship or UGC. Okay, so if you have your own fellowship, there is much good opportunity of getting into the institutions. Otherwise the institute will provide you the same amount as fellowship, so that is also fine. Okay, so once you appear for these exams, you will have to keep watch for all the different institutes. They will release their applications. Fill in the applications. So be it IISc, be it all the IITs, NITs, all the individual institutes, for example NCBS, then TIFR Hyderabad, TIFR Mumbai, all of these different institutes, they will release their own applications. Fill in the applications and then sit and wait for their call, ICMR, etc., etc. So you have to appear for interviews, and once you start appearing for the interviews, you'll become a lot more confident and more well versed with the whole process, and then you'll get calls from a lot of different institutes, and you'll have to choose where you want to go to. So yeah, all the best to you. Okay, right now the GATE results are out, so you also have given, yes, of course you have to give interview everywhere. So every place will ask for interviews. So in every place there is this multiple level exam, right? So first application, then they will select, then they will ask you to appear for maybe exam or direct interview, whichever it is. And some places there are 2 rounds of interview. For example, in NCBS there was 2 rounds of interviews. So that also you have to clear, and then you get selected. So that is how it is. It will, thank you, yes. Thank you so much. All the best to you too. Bye bye. Take rest. I'm ending the call.